Should I take over? Yeah. Okay. Okay, welcome everybody to the poster pitches. I would please ask the session 1 poster pitches presenter to join me here literally on stage or close to the stage. I will leave the stage to you all. Try to line up in the way that you remember your presentation were. There's not a chance to test your sorting algorithm right now, so please try to remember who was before you. And just as an indication, I will be strict and fair, so like I will give you a 5 seconds warning when you should be wrapping up, and then I'll cut you brutally or call for an applause once you should be done. Okay, I'm gonna move to this side so that you actually have a better view on the presenters, which is more interesting. And I think we are ready. Should I switch?
Explain this to them.
Yeah, I basically, they don't need a pointer, right? I change the slide.
You change the slide?
I will. Okay. Like, I possibly. Okay. As I said, I will be brutal and I will be changing the slides for you if you're not on time. So please to be respectful of the time that you got allotted. And we start with our first presenter, Ahmed Salah, with Doremitra, a curated data package for radiation dose response measuring transcriptomics.
Thank you. Hi, everyone. So if you work with transcriptomic studies and you want to compare your results with public datasets, you probably heard the same discordant tune that I did, inconsistent metadata even at the sample level within the same study. Therefore, we gathered in Harmony radiation transcriptomic studies and curated them with unified metadata annotation across all the datasets. And these datasets are now available by Experiment Hub as summarized experiment objects that are ready to play in full rhythm with bioconductor ecosystem for comparative analysis and biomarker discovery. Doremitra, as a one-stop shop for radiation transcriptomics, also come with user-friendly functions to help the users compose their own analysis without the headache of metadata noise. If you would like to know more about it, I will be happy to let you hear the full symphony at my poster. Thank you.
Thank you. Perfectly on time. Let me just give you the time to reset the timer. Because you were too fast, or like perfectly on time. Next presenter is Aitor Moruno Cuenca, SINGIST, an integrative method for comparative single-cell transcriptomics between disease model and humans.
Okay, so our poster is about a recurrent question that we always have in research and the drug discovery space, which is basically when we have a disease model, we always ask ourselves, is our disease model good in terms of how it's mimicking the human disease it's aiming for? And so you can kind of tackle this problem from multiple optics. One of these optics could be single-cell transcriptomics with bioinformatics. One of the problems, however, is that we don't have many methodologies to do this with single-cell transcriptomics specifically. And so for this reason, what we do is we introduce a new method, which is called SINGIST, which provides you with quantitative and explainable measures and also interpretable measures of how well are the single-cell transcriptomic changes of your disease model aligned to that of the single-cell transcriptomic changes of a human. And we also provide with a library for this.
Thank you. Next speaker is going to be Alex Cecchetto with latent structure modeling spatial transcriptomic data.
Hi, everyone. In my poster, I will present this generalized matrix factorization model that actually Levin this morning presented. And through this model, we are able to, through a linear predictor, to handle gene-specific effects, specific effect, and to capture residual variation through a matrix decomposition. And to handle spatial data, we build a spatial functional regression that the main idea is to use a basis function at different resolution to capture significant biological signal. And if you are interested to know more about this and to see a real data application, I'm at panel 4. Thank you.
Thank you. Next speaker is Ali Mustafa Anwar with Limrods, a hybrid method integrating empirical base and reproducibility optimized statistics for robust differential expression analysis. It's good that I had to read it so you can save a couple of seconds.
Yeah, hey. As we heard today, like in proteomics analysis, we have a lot of problem from batch correction to imputation, but also in the differential expression step, because people all, like in the most methods used, they don't have an optimization for maximizing the reducibility in the stat or the ranking of the proteins or the feature. This is why we created Limrods, and Limrods trying to maximize the reducibility by both precision and recall. And we tested Limrods against different DIA tools, which is widely used in proteomics, and also in 21 different data sets with different proteomics data set, and Limrods came out on the top of the list in most of them. Thank you.
Thank you, Ali. We move on to my GeneTomics friend. So Angelo Velle with GeneTomics, a powerful bioconductor package for multi-omics data integration and visualization.
Okay, so good afternoon. Today I'm going to show you some major updates we did to GeneTomics, our bioconductor package for multi-omics data integration and visualization. And GeneTomics is designed to detect the impact of genomic and transcriptional alterations on gene expression. And in particular, this year we added the possibility for users to split the samples in different groups and then to check whether there are genes that are differentially methylated or differentially altered in terms of copy number variations. And then the user can also check if these alterations are actually significantly impacting gene expression. Then we also provide a couple of case studies to show the potential of the package. So if you want to know more about this, come to poster number 7. Thank you.
Perfect on time. Okay, next speaker is Anna Bortolato with graph-based multi-omic survival and 2-class pathway analysis with the MOSClip-R package.
Good afternoon. I'm here to present a MOSClip that is an R package for topological pathway analysis on multi-omic datasets. As you can see, you can run MOSClip analysis on a whole pathway or after graph decomposition on its connected components that we call modules. The main purposes of MOSClip are to test for survival association of pathways or modules, or 2-class comparison. The package is available on Bioconductor, and it also includes several graphical tools for results visualization and interpretation. And if you're interested in MOSClip applications to a case study, don't miss the short talk by my colleague Anna Tanada later today. And for MOSClip functionality, visit my poster number 8. Thank you.
Perfect. Also with a double level of advertising. And now we have combining multi-omics factorization methods for robust biomarker identification, presented by Ivo Qui and Antonino Zito.
Hello, everybody. So we know that multi-omics data integration is really important for biomedical research, but there is a problem that there are many methods, we never know which one to choose. Essentially, all of them are factorization methods that produce factors that explain heterogeneity in the data. Some factors have different orthogonality, they differ in regularization, some of them are supervised, some of them are unsupervised. So to couple with this problem, we applied a new methodology which is based on normalized score from each method and produces a total variable importance as the cumulative ranks across the method. And in a multi-omics TCGA breast cancer dataset, by using this method, we found that the first biomarker was HER2, which is the most important predictor of positivity of breast cancer to this molecule. And now the omics playground is fully equipped with this model, and we look forward to that. Thanks.
Thank you, Antonino. Next speaker is Artur Manoukian with Voltron, a spatial omics analysis platform for multi-resolution and multi-omics integration using image registration.
Okay, so image processing is hard in R. It's not really impossible. So we came up with a package that we embedded a powerful computer vision C++ library called OpenCV, so that you can align adjacent tissue sections and serial tissue sections, multiple spatial omics that you capture from tissue sections. But then you realize that the results of these analyses are not really captured in the existing frameworks, so we came up with our own, and we came up with additional delayed array-based dependencies to capture large metadata sets and large pyramidal images. Check out the poster if you like to learn more. Thank you.
Perfectly. Thank you. Our next speaker is Claire Rioualen, interoperability of bioconductor packages with the Elixir research software ecosystem using the EDAM ontology.
Hi, so if you've ever submitted a bioconductor package, you've probably used BioCV's terminology to tag it, which allows to organize and navigate the overall collection of packages. However, a couple of years ago, I met Maria, and together we thought we could improve the situation by using the EDAM ontology for the annotation of packages. The topics are related to the kind of functions they perform, the kind of data that they process or produce. And so the overall objective of this work is to improve the interoperability of the bioconductor packages across a wider ecosystem of resources that are available. And so now we would really like your inputs, the package developers, package users, about what we've done so far and what we plan to do next. So if you're interested, please visit poster number 12 or check it out on Zenodo with the QR codes. Thank you.
Perfect. To the split-second level. Next speaker is Dani Mukesha, metabolomic signatures and machine learning models for distinguishing Alzheimer's disease and dementia with Lewy bodies.
This will be a side project of my PhD, and I will be more focused on metabolomics and also specifically in Alzheimer's disease and other type of dementia. As you all know, Alzheimer's disease is a complex and multifactorial disease, and complex disease cannot be explained by genetics alone. So metabolomics is a beautiful place to look at other influences that are going to contribute to the development of a disease. So in this research, I focused on identifying new biomarkers in metabolomics, and I was able to use machine learning and different models to identify up to 67 metabolites that can accurately differentiate between Alzheimer's disease and Lewy bodies, which are the most predominant type of dementia. And if you are interested to learn more about metabolomics and how to apply machine learning in targeted metabolomics, you can come to see me at poster number 13. Thank you very much.
Thank you, Dani. Our next speaker is Eduardo Filippi with bringing omics together with Movida, multi-omics visualization, integration, and downstream analysis.
So we all know that working on bioinformatics data, especially maybe when working with a collaboration partner that doesn't have a lot of bioinformatics expertise, is difficult. We found ourselves in this situation, and we thought that maybe we could make the best out of it by developing an application that in a multi-omics setting, which is the one where we are now, with working with transcriptomics, proteomics, and metabolomics, could solve our problems, and maybe also yours. And the idea is that we want to be able to do pathway-based integration, map the results on cake pathways, and also be able to kind of map the relationships between layers with some network analysis. We also wanted to be able to easy to integrate in our workflows and to be, export data and plots to make it reproducible. If you have ideas, comments, and
you should
anything,
check its
come
poster.
to me.
Not only because I'm also involved in that, but okay, our next speaker is Eugenie Loman, Analysite, a GUI for automated post-clustering analysis of cytometry data.
Good afternoon, everyone. I'm really happy to speak about the project I had with my labs. It's concerning post-clustering in cytometry analysis. So cytometry researcher usually doesn't know how to handle post-clustering analysis. So for that, we built a Shiny app application that handle the creation of reports for quality control, differential abundance analysis, and differential scale analysis. For that, I used Shiny, Quarto, and also EuroBioC. So if you want to know about my project, please come to my poster, numero 17. Thank you.
Thank you. Next speaker is Giulio Benedetti with Mia Dash, a web app for reproducible and interactive microbiome analysis and visualization.
So hello, everyone. Data sets in microbiome research are expanding quickly, both in volume and the number. And there are tools available, like Mia and Philosec, to study those. However, it's still harder for people with little knowledge in programming to analyze and visualize this kind of data. Therefore, we developed a microbiome analysis dashboard that emerges from two packages, actually, Mia and IC, which is, I think, a great example of interoperability in bioconductor between two packages. It's also helpful for people who know programming, because you can replicate some analysis workflow from this package as a code. And it's really interesting that it's stamped 2 years ago in EuroBioC in Ghent, so it has exactly 2 years. Thank you. Visit my poster at number 18. Bye-bye.
Thank you, Giulio. Next speaker is Guillaume de Flandre with PS Match, a bioconductor package for handling peptide spectrum matches data.
Yeah, so the processing of mass spectrometry-based proteomics data can be roughly divided into two sections: the identifications of peptides and their quantification. And I'm here to present to you the PS Match package, a part of the Alpha mass spectrometry suite, and which solely focuses on the identification part. So it allows to load, manage, and handle your peptide spectrum matches data, but it also allows to annotate and visualize MS2 spectra with or without variable or fixed modifications, but also store peptide-protein relationships and visualize these relationships. So if you're at all interested in how or why you should analyze your identification data, don't hesitate to come to our poster at number 19, or scan the QR code that brings you to the preprint, which explains PS Match. Thank you.
Thank you, Guillaume. Our next speaker is Himanshu Saraswat, applying BioC packages to identify genes and pathways relevant in multi-case multiple sclerosis families.
Hi, everyone. Multiple sclerosis is a complex disease. So to identify the causes of MS, there has been a lot of GWASs, but they have found common variants, not the rare variants. But to identify rare variants, we would need lots and lots of population. That is where the families come into play, as families have these rare variants which are exaggerated in the family through the Mendelian inheritance. And I looked at 6 families in which I found 223 coding variants in around 215 genes. And then I used all these different tools, which I have mentioned there, to see if they are converging into any single pathway or multiple pathways, which we can look further. So we have identified a few pathways which are related to neurogenesis and exon, actin binding, and all these pathways have been implicated in one way or another with other neurological diseases, and it brings to us that there are something that is in play regarding these things. So we want to identify them more, looking into a bigger case control cohort and see if these pathways are expressed there as well. For more, meet me at poster 20. Thank you.
Perfect, thank you. Our next speaker is Julia Mir-Pedrol, bridging bioconductor and NF core toward portable, reproducible, and scalable biological pipelines.
So have you ever had to analyze a large data set and you wished you had a reliable way to run your code without worrying about scalability? Or did you have to reuse that same code or share it with a collaborator only to find out that it breaks due to different environments? My name is Julia, and in my poster I will introduce you to Nextflow and NF Core. These provide solutions to these problems. They set standards, they allow you to build reproducible and portable pipelines, and bring together an active community of contributors. Some of the most used NF Core pipelines rely on bioconductor, and moreover, we have modules wrapping some of the most popular bioconductor packages ready to be used in any Nextflow pipeline. So if you are curious and you want to learn more or want to bring your perspective on how to bridge the bioconductor and NF core communities, make sure to visit my poster.
Thank you, Julia. Our next speaker is Katerina Matejkova with SAFRA, splicing annotation for aberrant fraction analysis.
Up to 14% of germline pathogenic variants affect mRNA splicing, which can be detected by RNA sequencing. However, current tools lack the capability to quantify the aberrant fraction, which is necessary for variant classification in alignment with ACMG-AMP criteria. Therefore, we decided to create SAFRA, which is a tool that annotates the splicing events, detects the aberrant splicing events, and finds their matching canonical counterpart. And this helps us to quantify the aberrant fraction. This is a bioconductor, or hopefully it will be bioconductor tool that uses tidiomics syntax, and it can be used in your workflows that you use in diagnostics of Mendelian diseases. So if you are interested in how you could use it in your diagnostics workflows, please stop by my poster. Thank you.
Thank you, Katerina. Next is Samurai shallow analysis of copy number alterations using a reproducible and integrated bioinformatics pipeline with Sara Potente. So find the name.
Sorry, thank you. Hi, I'm happy to present today my pipeline that is called Samurai. It is a Nextflow-based pipeline, and it is used for the analysis of copy number alteration from shallow whole genome sequencing data. And in Samurai, we have a sub-workflow specific for solid tissue analysis and liquid biopsy analysis. Now we are expanding Samurai with additional modules and also a user interface in order to avoid the command line of Nextflow pipelines. And yeah, if you want to ask some question or discuss further, I'm happy to talk with you at the poster session, poster number 30.
Thank you, Sarah. Next speaker is Thomas Bormann with orchestrating microbiome analysis with bioconductor.
Yes. Okay, let's start with the exercise. Please raise your hands up like this. Yes, I have time to wait. Exactly 50 seconds. Yes. So how many of you 50. Like microbiome data science?
Keep your hands up.
All right. So we have developed this ecosystem of for microbiome data science based on this summarized experiment data container. And then we have this online book, photo. Yes, it has all the answers to your questions. And if you think that I promised too much, please come to my workshop tomorrow, or the poster. You can get a T-shirt there in the workshop.
That's a surprise. Okay. Okay. Thank you, Thomas. Our next and last speaker for this session is Yosra Berkwayel-Dahour, refining transcription factor enrichment analysis with the upgraded TFEA chip.
Yeah, thank you. When we do an RNA-seq analysis, we end up with a list of genes that are significantly regulated. And one question that we may ask is which transcription factors are modulating those genes. So to answer this question, we can run a transcription factor enrichment analysis using the FEA chip. The FEA chip works by associating regulatory regions to genes, checking their overlap with ChIP-seq experiment from REMAP, and then testing whether those targets are overrepresented in the group of regulated genes. So here, the association between regulatory regions and genes is key, and in the new version of the FEA chip, we have changed that from GeneHancer to the new ENCODE Re2G database, which performs better compared to other state-of-the-art tools. And we have also added other functions that make the package more powerful and user-friendly. Thank you.
Thank you, Yosra. I would like to, again, maybe, in the spirit of what Thomas did, I would like to invite all the speakers of this session to stand up, and you can join me in thanking them again for the bravery to hop up on this stage and then have a nice poster pitch. Go see their posters.